Hi everyone, thank you for being here. My name is Gemma. I'm a fourth-year medical student at Virginia Tech Carilion in Roanoke. Today I'll be talking to you about my project titled Adverse Birth Outcomes in Drinking Water Quality, a retrospective analysis using West Virginia Medicaid data. So this study focuses on Appalachia. It's the region highlighted in white on the screen here. It ranges from Mississippi all the way up to New York, and it's a geographic and a cultural region of the US that has been recognized by multiple organizations for demonstrating persistent disparities that require further research. For this study, we're looking specifically at water and health. And water access challenges in this region are due to several factors, most likely topography. It's a very mountainous region, underinvestment, pollution from coal mining, just to name a few. So the research group that I work with at Virginia Tech recently conducted this review and meta-analysis of the region, and they found that there are very limited amounts of studies within the region that focus on water, and even fewer that focus on public health. So that was one of the main reasons we decided to do this study, is just the need for more research in the realm. So the overall research objective of this study is to investigate adverse health outcomes in maternal and neonatal populations in West Virginia in relation to estimated access to safe drinking water at a county level. For reference, this is the state of West Virginia that we're focusing on for this study, and these are the counties. This study is specifically looking at maternal and neonatal outcomes, and one of the main drivers behind this is looking at infant mortality rates in Appalachia as a whole versus the US is 7.1. And then looking at the different regions, subregions of Appalachia, Central Appalachia, which is where West Virginia is located, was one of the highest regions of infant mortality. Previous work in this space has shown that specific safe drinking water violations are significantly associated with adverse birth outcomes. It's also shown that there's an increased risk for preeclampsia with higher water sodium content. And finally, linkage of infant health outcomes from birth records with drinking water contaminant measurements in Pennsylvania showed negative effects on birth outcomes. So for this study, we hypothesized that access to utility supplied drinking water with few or no monitoring or water quality violations among Medicaid recipients in West Virginia will be associated with a reduction in the odds of preterm birth and low birth weight. And specific aims of this study include synthesizing multiple publicly available data sets to estimate water source availability and socioeconomic data, and to look at the statistical associations between access to utility supplied water and the outcomes of birth weight, low birth weight, and preterm birth. So getting to some of the data sources that we used for this study. So the primary outcome data source was Medicaid data. Medicaid's a joint federal state funded social welfare program that provides health insurance to low-income families. They use a fee-for-service model where there's a direct link between the service, the payment, and administrative data, such as demographic data. So that's what we capitalized on in this study. And these are some of the variables. In terms of other data sets used in this study, we used the Safe Drinking Water Act to look at health-based water violations. This act was established in 1974 in order to protect consumers. From this data set, we used federal level data, which looked at water system detail and violation reports from the Safe Drinking Water Information System. We looked at the time frame 2010 to 2019, and some of the variables were water system name, ID, county served, population served, number of violations, and types of violations as well. And then other sources of data used, we used the vital statistics from West Virginia Department of Health, and this was data on population health, mortality, fertility, and other vital events. And then for demographic data, such as income, county population, age, education, we use US Census data for that. Okay, so this is the original analysis plan. The plan was to take the Drinking Water Act monitoring data and the Medicaid data. Drinking Water Act monitoring data was used to estimate the number of health-based water violations at a county level. and this was used to estimate the maternal exposure to contaminated drinking water. The Medicaid data was used to estimate the adverse maternal and fetal health outcomes, and we were looking at low birth weight and preterm birth. Linkage of these two data sets by county was then the source allowing for analysis of the associations between birth outcomes of interest and drinking water violation data. Okay, so looking at some of the results, looking at the Medicaid data, we found there are about 210,000 Medicaid records. About almost 7,000 of these showed omission for childbirth during 2019. And about 411 of those 6,700 were preterm births, as determined by ICD-10 admitting diagnosis codes. This is, if you take the prevalence, it was about 6% of births, which was much lower than the US mean. Next, we looked at birth weight and found that there was only 52 records of the 210,000 that had a birth weight recorded next to them. Looking at this a bit more, we think what might have happened is that these low birth weights and preterm birth records were tied to the infant health records and not the maternal health records. So currently we're trying to figure out the best way to incorporate the Medicaid data into the final analysis. But for now, the rest of the analysis I'm going to show you was done with the vital statistics, census, and safe drinking water act data. So looking at vital statistics data, there were 18,090 births in West Virginia in 2019. 1,775 were low birth weight babies, and that was above the national average, 9.8 versus 8.3%. And then looking at the water quality data, there were almost 9,000 drinking water violations from 2010 to 2019, and 571 of those were health-based. Okay, so here's some of the preliminary results. This is from census data for counties across West Virginia. The average income across counties was $45,200. Looking at the education levels, the greatest proportion of women who gave birth in 2019 in West Virginia had either a high school diploma or some college. And the greatest proportion of women who gave birth in West Virginia in 2019 were between the ages of 20 and 34. The average household size was 2.44, and about 3/4 of every county was served by utility water. The average number of health-based water quality violations per county was 10.4, and the proportion of violations per county for a population served by the utility water was 14.2. Okay, so then looking at the relationship between low birth weight and water quality violations, we found that there was a weakly positive association between the two. So we did expect that they would be positively associated, that with the increase in proportion of water quality violations, there would be an increase in the proportion of low birth weight births. However, looking at the model here, so we plugged the data into a linear regression and found that that same positive relationship continued to exist. However, there was not a statistical significance present there. Next, let's look at some of the different variables that can impact low birth weight. So initially we were talking about just water violations, but some of the other things we looked at were median income, age, education. So looking at median income for households in West Virginia per county, we found that as median household income increased, the proportion of low birth weight births per county decreased, which is also what we expected. Looking at this in the model, we found that that same negative relationship persisted, which we expected, and that this relationship is approaching statistical significance at a p-value of 0.05. Some of the other factors that we considered as potential confounders were proportion of women who graduated high school. This also had a weakly positive relationship that was not statistically significant. And we also looked at average household size, which was a negative relationship and also not statistically significant. Finally, we did a full model including the relationship between low birth weight and water quality violations, accounting for the different confounders that I described, income, education, and household size. We found that the weekly positive relationship between water quality violations and low birth weight continued to persist, as well as the inverse relationship with median income and low birth weight. So this project is still ongoing. We're gonna go back and take another look at the Medicaid data, figure out the best way to incorporate that into the analysis, and hope to have a paper out next year. I'd like to acknowledge the Virginia Tech research group that helped me with this project, Dr. Cohen, as well as other colleagues in the Department of Population Health Sciences at Virginia Tech. Thank you.
Questions? Good.
I was curious, when you're looking at this research, you don't see a lot of these correlations that you thought you might see. Do you have rationale for that? Do you have reasons you think they might not have been as strongly correlated as you thought they might be?
So you're talking about like the water quality?
Yeah.
I think that there's a lot of factors that go into play with that. So we were able to estimate the percentage of the county that was served by utility water, but the percentage of the county that's not served by utility water, we don't know if that's like spring water or well water. So we weren't able to account for those things that could be affecting birth outcomes as well.
Questions? Yeah.
Thanks for the talk, really interesting. I guess it's a little unclear to me with your model, like what your betas are, how do I interpret the betas here? It's like, and what's the public health relevance of something that's like 0.0001? Maybe you can just talk me through like how would I interpret that? And then is it a relevance to public health, or there's something, these are like, seem like they're very small associations, maybe?
Yeah, so these are still preliminary results, so we're gonna take another look at it, but it is very small and it's not statistically significant. So at this point, I don't have any specific recommendations based on the data.
Nope. Okay, so thank you. Okay. Next presenter, Andrei Badilla-Aguilar.
Hi everyone. After 4 long days of conference, here I come to talk about cow poop. Very exciting, isn't it? I'll add a little bit of engineering and math to it to make it even better. So I'm a PhD student at the University of Notre, and my research focuses on the transport and fate of antibiotic-resistant genes from cattle manure in flowing systems, specifically rivers and streams. Just a couple of heads up, I will present at the end of the presentation 2 QR codes, so for folks who are interested in these topics and educational modules, I will highly encourage you to get your cell phones ready. And I will present sneak peeks of my main project and some scattered results, and hopefully we can get some great conclusions at the end. So to start off, despite claiming more than 1 million human deaths live annually, the antimicrobial resistant crisis extends beyond human health settings, including hospitals. For example, the animal raising husbandry industry is the main consumer of antibiotics worldwide, making farms suitable environments for antimicrobial resistant genes and genetic mobile elements to emerge, transfer, and persist. In fact, some agricultural activities have been recognized as highly contributors for the AMR burden in the environment. Particularly, the land application of manure stands out as a really important source of contamination for surface waters. And so what farmers do is that they take the manure that their cows generate, they put it on the field, they fertilize their soils, and so those crops will be used to feed the cows again, which is great for them. However, after rainfall events, contaminated runoff will carry all these pollutants, including ARGs and bacteria, to rivers and streams. What is happening in the stream, we still don't know, and there is a plethora of mechanisms that could be going on. And specifically, in order to tackle this problem and generate better predictive models, we need to understand how these ARGs and how these bacteria are moving and what their fate is in the environment. And so to tackle this problem, at Notre, we use an increasing scale approach. So we're very fortunate to have a couple of facilities that allow us to play with factors and understand better the transport of pollutants. So for example, we have the experimental mesocosm facility where we have 24 fluid mesocosm units where we can spike in pollutants, see throughout time how these are being removed from the water column. But the rockstar here is the linked experimental ecosystem facility. So we have, artificial rivers fed with groundwater 365 days a year, 24/7, and we can do wide range of experiments here. And also we perform real world experiments as well, although it is not part of my project, and I will focus on these two settings. And so to answer the very first question that we had is how this ARGs transport in a flowing system. And so to tackle this, we grabbed one of these artificial rivers and we went to a farm, grabbed fresh manure, and spiked in this fresh manure into this river that was continually flowing. We performed this for 3 days. And so we took sample when we reach a steady state. How do we reach a steady state? Where we use a passive tracer to measure a steady state across the stream. When we knew a steady state was achieved, we collected the samples, took them to the lab, and quantified a raminofecal marker, BACAR, and 3 antibiotic-resistant genes. We then fed linear regressions to our data, and then feed up a model, the stochastic model. If you want to hear more about the model, I will be happy to touch base about it. Otherwise, it will take us around 3 hours to understand it. But what we want to know here is that this model allows us to get 2 temporal rates. And these temporal rates will explain how our targets or pollutants in the river are being removed depending on interactions on the subsurface, which is the bed of the river, and the surface, which is the water column of the river. And so this work was published some months ago, and this is just a subset of the data. Interestingly, we didn't know what to expect, and this is pretty novel. We found that these targets are being removed exponentially from the water column, and so they disappear very rapidly. When we feed the model, this model told us that this removal is being driven by stream bed interactions occurring at the bed of the river, probably at the benthic biolayer zone or the hyporheic zone. And so when we have really well-characterized rivers, we can actually use this tool to kind of predict how these ARGs will move in real environments. Although we are aiming for this work that the rates will be using a more complex model that will take into account several biological factors, although they fit very well what we do in our artificial rivers. And so now we know how fresh manure borne ARGs behave. How about other types of manure? It turns out that farmers can store manure for months or years, can use manure from anaerobic digesters. And so we went to the field again in another experiment and grabbed a sample from, again, fresh manure and from a store source that was considered store/treated. And so this manure was being in the pond for like 3-4 months. We brought the manure out of it, and we changed a little bit the experimental setting here. Rather than doing a single stream for 3 days. We did 4 shorter streams on 2 different days. And we did the similar approach, quantify the same genes and fitted the same model. Our hypothesis here was that the removal is being driven by target. It may be a host. And so we expected to see kind of the same removal. Surprisingly, we didn't even see removal in some of our targets on store manure. We obtained positive slopes, which was very interesting for us, including significant differences between the removal of 2 ARGs and at the fresh and store manure, suggesting that storing manure actually enhances the persistence of TET-Q and MEF-A, these 2 ARGs that we measured. And so when we fit the model, we actually saw the same result, that what is happening at the subsurface or the stream bed is what is driven the removal, but these removal interactions are diminished in store manure. We don't know why, and that's something that we are working on to tackle. And since we now know that the stream bed is playing a big role and that biofilms grow at the bed of the rivers, how do we know what's the influence of biofilms here? So we follow a very simple approach. We 3D printed some slides that we then took to our artificial river and colonized them with biofilms. So we put them on the water column for 3 weeks. And so we use the other great facility that we have, the experimental mesocosm facilities. And we did 2 experiments, a short-term one when we spiking the cattle manure in this experiment, start the recirculation, and sacrifice one of these slides along with a water column sample for every 5, 10, and 15 minutes, and then a long term, because we also wanted to know what is happening at the long term. We grabbed the samples to the lab and we quantified the concentration of several ARGs, and we calculated attachment and detachment rates, and you will see why. So this panel, concentration on the y-axis and time on the x-axis, we saw a rapid attachment of these ARGs to the slides. This is the results from the slides. And so all of this fitted an exponential model perfectly. We saw this attaching very rapidly. And however, when we took a look at the long-term data, we saw detachment. And so this is very interesting, although not as quick as we saw in the very first experiment. We saw detachment, which could be explained by senescence of the biofilms in the slides and other factors. Some conclusions from our work: I think we obtained really good experimental novel rates that will be used to feed up other models, and that will allow us to track and hopefully tackle in the future the advance of AMR in the environment. I think the matrix matters. We still don't know what kind of interaction or metabolic adaptations are occurring during treatment or storing manure. And we're further working to see if the attachment of these ARGs to the biofilms follow any sorption isotherm, or what is the mechanisms behind this. And so future we will look into how do these biofilms acquire the resistant genes. Are bacteria being recruited to be part of the biofilms? Is this external DNA being just a structural part of the biofilm? That's something that we still don't know. We are working to figure it out. And some of my projects will include looking at the external and internal DNA and disentangle how these 2 types of ARGs are being removed. And so last but not least, I would like to highlight some work that we did for several months to create an educational platform, an educational video about antimicrobial resistance, agricultural settings, and the impact on human health. So we spent a lot of time doing these videos, so I highly encourage you all to take a look and feel free to use them. That's for everyone. We went back to the 90s, so if you missed the 90s, I will definitely go and check them out. In the didactic platform, we have educational content, including quizzes and a video game that is actually pretty fun. And so, yeah, with that, I would like to acknowledge my PI, Dr. Kyle Vivian, the co-PIs of this work, Diogo Bolster and Jen Tang, and of course my wonderful collaborators, because that was a lot of field work. And yeah, I will address any questions that you all might have, and thank you.
So, questions?
Thank you, it's very fascinating. Sort of a couple of things actually, just regards to, just trying to wrap my head around. So you're looking at the rate of degradation of the genes. So does that mean when you are trying to control the load of, so firstly, do you then try to standardize the ARG load burden that you then introduce into the river stream? So how would you, so does that mean you then have to check what's the concentration at the start? And do you then do this by qPCR? Yeah.
That's a great question. We did a lot of trial and error because we had no idea about the concentration of the antibiotic resistant genes. And actually we were constrained. You know, sometimes you go to the field and you want to find a lot of stuff, but at the same time, you don't want to find these things in the environment. And that was our case. These cows were really healthy.
Yes.
And the rest system were really short in terms of number of genes. And so, yeah, we did a lot of experiments to know how much manure we would put in the rivers. And we also had to know that you shouldn't go above the EPA limitations of E. coli in terms of surface water. So that was a lot of work to calculate how much you should add in order to keep your E. coli levels down, but also have a signal when you collect the sample and do the PCR. But yeah, it was a trial and error thing.
Sorry, just a clarification. So I'm a bit geeking out on this because I do like AMR. So does that mean that for every experiment you work out what is your T0 or your inoculum when you inject into the river and then you track it down?
Correct, and it is very high on cattle manure. So we usually work with 1 gram per liter solution to do our spiking, and that is more than enough to get a signal.
Thank you.
Yeah, really interesting. There's a lot of data. Again, trying to wrap my head around all of it. I guess my biggest question is this was all molecular work, right? And you're talking about ARGs, but ARGs are carried by bacteria. And you mentioned biofilms a little bit, but I'm curious, how do you see bacteria mediating this? Did you guys do any culture-based work to complement this? And if you talk a little bit more about that, that would be really interesting.
Yeah, it is actually pretty difficult to, at least at the culturing level, disentangle what the host are. We have another, I didn't include this, I'm co-leading that project, but I'm not the lead on that one, where we actually included the culturing side on the mesocosm experiments. And just what I was most surprised about is seeing bacteria carrying beta-lactamases being still viable after 10 days of spiking in water with low nutrients. So it's definitely something that we're working on that data right now, but that's a sneak peek that it takes a while for them to at least decay in terms of viability. So yeah.
I had a very quick question. I was curious, I know you said you didn't know exactly what the stored feces had been treated with. Do you have any idea, or is it just really, it was treated?
It's an anaerobic pond. So that is a great question. In our first experiment, we used fresh manure and we found the ruminant fecal marker to be super concentrated, which is something you would expect. When we went to the field and grabbed this treated store manure, we didn't find it. So the ARGs were still there, but the ruminant fecal marker was completely removed. And these are anaerobic ponds, and they use them, to store the manure, but the farmers don't know that there might be some degradation and treatment occurring. So that's what we also call it the treated manure. Yeah.
Thank you.
Next question.
Very interesting talk. So if you saw a lot of interaction with like the stream bed because of potential biofilms, then in a real situation where you're looking at like potential runoff from farms into rivers, would you expect there to be a lot of runoff, or would you expect a lot of the ARGs to get kind of stored by the biofilms, in the ground, and then not actually go into our waterways?
Yeah, that's a great question. I think there is enough. I think there is enough evidence for folks to work with soil science to support that a lot of the ARGs will be kind of like attached to soil particles, either for bacteria attachment, absorption, and other mechanisms. But there will be some runoff if the guidelines are not followed in terms of manure application. The only problem here is that guidelines are very lax in some states, although we are hopefully making an effort to change that in the future. But in some states, the guidelines are completely lax and farmers do whatever they want with the manure. And yeah, I would expect, even though there is some absorption occurring in the soil, there will be some leachate rate getting to the water bodies.
Thanks, Andrei. So I have a question about, so you had biofilms. I was curious about like the flow rates. How does like external factors like temperature, pH, like the flow rates in terms of biofilm attachment, in terms of biofilm building, how does that affect in terms of like the transport and fate of the resistance?
Great. Yeah, we actually do this at 250 RPM. The recirculation is at 250 RPM, and this might be a detail that you guys don't have to know, but we did this experiment, and 1 month afterwards, the guy in charge of facilities bought some devices to heat up the mesocosms. So we would have done some interesting temperature experiments if we would have known that this guy was buying these devices. So that's something I would love to look into, and there is a group in Germany that does a lot of work in terms of temperature and biofilm absorption attachment of antimicrobial resistant traits, and I think they didn't find any sort of relationship between attachment and temperature in the last paper I checked, but I'll be more than happy to touch base about that.
Okay, so thanks, Andrei. Next presenter, Rehnuma Haque. How are you? Good. Thank you.
So, cow poop to food. Please bear with me. And hello everyone, good afternoon. I'm Rehnuma Haque Sarah. I'm an assistant scientist and working at icddr located in Bangladesh. So today I will present on the microbial contamination status and associated hygiene behavior of primary caregiver of the children of age between 6 to 24 months. So these children are at their weaning age and live in low-income settlement or slum. So in this picture, you can see it is the largest slum in Dhaka, Bangladesh, which is the capital city of Bangladesh, and the slum name is Carilion. So I would like to kick start by acknowledging the invaluable contribution of our diverse team members. Our team's expertise spans various fields, including anthropology, behavioral scientists, laboratory scientists, statistician, physician, infectious disease modeler, and we also benefited from the collaboration of government representatives from the Institute of Epidemiology, Disease Control and Research in Bangladesh. So, what people definitely know, this is the typical slum scenario. The image on this slide depicts the challenging hygiene condition in Carilion. The densely packed tin-shaded houses provide minimal protection and narrow, poorly drained often roads are wet, and household entryways are surrounded by waste, which reflect that improper waste management. And also there is shared kitchen and shared toilet, which serve multiple households, are often poorly maintained and with lack of basic sanitation. Also, the proximity of the kitchen to toilets are very close, that remains concern about cross-contamination. So this condition presents significant health threat to the user. So previous research on this topic has primarily focused on high level of E. coli contamination at household and the child weaning foods. However, there is a research gap in other foodborne pathogens that contribute to diarrheal diseases. And this diarrheal disease during weaning food in the children plays major critical factor like enteric infection that leads to co-infection and malnutrition and increased fatality. So therefore, we need some research to understand the other pathogen diversity. So there are a few hygiene intervention trials happened in Kenya and Nepal, but still Bangladesh has lack of this kind of research, despite Bangladesh has highly dense populated country and climatic impact on the growth of pathogen diversity and also to integrated research practices. So we aim to do study design on 3 phases. First, formative research, and next co-design and hygiene intervention and feasibility trial. So now I will just present the preliminary findings of our formative research that aim to identify microbial contamination status in domestic surface, child weaning food and drinking water, as well as explore the domestic hygiene practices of primary caregivers in an urban slum. So for data collection, we utilize several tools to capture a comprehensive understanding of both microbial contamination and their hygiene practices. The multi-method approach allowed us to explore the socio-demographic, household environment, food security, and hygiene practices. And we considered the capability, opportunity, and motivation that Combi behavioral model to understand their total hygiene behavior. So here we initially collected the domestic swab, drinking water, and child weaning food, about 193 samples, and we did structured observation for each household from morning to afternoon. It is like 8 to 10 hours, and in the next slide I will tell what we try to see during our structured observation. We also have done some semi-structured interview with the primary caregiver of children aged between 6 to 24 months, and there is a group like first-time mothers. They are quite concerned about their children hygiene, and second time or mother of multiple children. And we also asked the adult caregiver like grandmother. And we did also key informant interviews with slum leaders, healthcare providers, the implementer from government and non-government partners. So during our observation, we have looked for the pre-identified behavior, identified behavior. So I will not go through this all behavior, but our primary goal is to identify the food handling, hand hygiene, kitchen hygiene, overall their household hygiene compound situation, and child feeding practices. So we collected these samples. Actually, these are mostly children food samples. So there are lentil soup, semolina, some rice pudding, and drinking water from their feeder bottle, and swab from the area where they store their food, like shelf. And we have done tested of these 12 pathogens via RT-PCR method. And Out of 32 households, we found 29 households were contaminated with at least one pathogen. And this table shows that across 97 food samples, 32 drinking water samples, and 64 domestic surface samples, among these, drinking water samples are particularly safe. But the domestic surface samples are pretty contaminated, up to 80%. And food samples are contaminated is about 33% for at least for one pathogen. And if we look the specific pathogens, the Clostridium perfringens, which is pretty toxic for enteric disease, came out across the all samples, and also rotavirus. Vibrio cholerae, Bacillus cereus, and Staphylococcus were found in both food and domestic surface, and Shigella was found only in the domestic surface. And we couldn't find other pathogens like norovirus group 1, 2, E. coli 0157, which is the most toxigenic E. coli, and Salmonella. And if we particularly focus on the food items like cooked food, that refers to item consumed within 4 hours after cooking. There is a stored food are kept for more than 4 hours. And reheated food is food that has been stored for more than 4 hours and then reheated at least for 2 minutes and before the consumption. And also there are snacks which are typically instant food purchased from the nearby vendors. So here we see that contamination status is higher in stored food. It's about 54%, followed by reheated food is 40% and snacks is 36%. And comparatively cooked food are safe with 19%. So we tried to reasoning this high contamination with their behavioral practices. So there are only few. We are still analyzing our data. So we found that during cleaning, chopping surface and equipment, like all their cleaning equipment, are not cleaned before use. So they cleaned once they're using it. Used utensils are stored unwashed from morning until lunch or dinner. And surface are not cleaned after children urination or defecation. Surface means domestic surface. Even soiled baby pants are stored indoor for collectively clean maybe next day or maybe day after. And during cooking time, we found that 2/3 of households have kitchen within 5 steps from their toilet. And cooking is typically done once a day, mostly in the morning time, and kept it all over the day. Storing practice came out like store food is stored on open shelves or under the bed. Reheating, they only reheated the food that had been stored in the refrigerator. And feeding babies, like baby are often fed by their hand, and mother are wipe their mouth by their wearing clothes. So there is no separate towel or napkin for baby cleaning. And currently we are trying to mapping this behavior under combi determinants in development of an intervention. And we are also considering or coordinating with the health team, hygiene team, and food safety authorities in Bangladesh. So actually it's not an actual conclusion, but conclusion of my slides. So the study confirmed microbial contamination in domestic environment and linked to suboptimal hygiene practice. And it also highlights the urgent need for intervention to improve home hygiene in urban slum. So after question and answer session, if you want to know much more, because we are in very preliminary stage, please you can connect with me through this QR code. And definitely I would like to acknowledge the funding from Beckett Global Hygiene Institute and travel scholarship from UNC Water Institute. And ICDDRB thanks its core donor for their ongoing support. Thank you very much.
Questions?
Sorry, thank you for your presentation. Maybe you explained and I missed it, but how do you explain such high concentrations in cooked food?
Yeah.
Like if they boil it, everything should be killed, and you still find quite high concentration, so it's reintroduced after the food is cooling down, or you
Yes.
measured on the utensils?
Thank you for the great question. So, our food sample collection method was depend on our observation windows. So we were waiting in the household and observed their cooking practices. And it was between 4 hours after cooking. And we took the leftover food from the child plate and analyzed it. So I'm not sure it's still that much hot just after cooking, or it's 4 hours like before end of this 4 hours. So we have to check, but I will definitely cross-check this. Thank you.
Hi, thank you, good talk. I was going to ask about this as well, but just on the same point.
Sure.
Obviously, if you're using PCR to detect all of your pathogens, PCR is going to pick up DNA from cells that aren't viable as well as viable cells. So I just wondered if you had any thoughts on, the overall infectivity of each of these sources, rather than just numbers of genes. Makes sense.
Thank you. I'm taking it as a comment because we have group and the laboratory scientists led this thing. So I will pass this comment so we can do it more in-depthly. Thank you very much.
Next question? Well, side I have my question.
Sure.
I didn't think so. You observed the households for a while, yeah? Didn't they change their behavior because they're looking at them?
Thank you for this question. Yeah, there is a doers and non-doers thing. So we initially we try to visit them 3-4 times, so they are quite familiar with us and they don't feel like outsider is sitting and observing them. So this rapport building helped to, I believe, that non-doer activities.
Hi, Papa. Quick, just a question in regards to the season. During what time of the year did your sampling? Because I was wondering, especially like you're saying, the other noroviruses and E. coli, they had some seasonal variation as well. And on the seasonality, do you think that also will impact on behaviors in the households?
It's an excellent question.
Can I jump question? So I don't know humidity in place, but humidity, if it's a dry season and humidity also may impact, yeah?
Okay, thank you. We always try to understand the seasonality impact, but unfortunately it's a one-time sampling, but we do collected the humidity and ambient temperature in this household. Also, we had some data on the food samples. So we collected data on the by food thermometer, and but I didn't analyze yet, so I didn't show it. So for our next phase or our trial, we are considering to catch the whole season, like 2 seasons, hot and cold season. So these data are from October, like September to December. So Bangladesh is still tropical country and temperature is quite high, but we are trying to see the May-June data in our next phase. Thank you for bringing this.
Thanks for the presentation. I guess I'm curious about the Vibrio cholera data. Was there like an ongoing outbreak of Vibrio cholera during this time in the community? Because some of the PCR assays were developed for like child species clinical settings and will pick up other Vibrios in the environment. So it's kind of curious, do you have, is there like clinical data to support there's like ongoing transmission of Vibrio cholera at that time?
Thank you. So in Bangladesh, Vibrio cholerae is the home of Vibrio cholerae. So even in our wastewater surveillance, we found high peak of Vibrio cholerae. So that time, I don't remember is there any outbreak, but we are from the diarrheal disease research center, and we have big hospital for diarrheal disease. So majority of patient came with Vibrio cholerae. They diagnosed by Vibrio cholerae infection.
Next question? No? So, thank you. Very interesting. So, our session is done. So, if you wanna, people, just go and have some fun with some wine and see the posters over there too. Thank you all.
When I saw the presentation now, it's how lucky I am of not having been born in a place like that.
I'm an anti-colonizer.
The photos and the figures, oh my gosh.
I think the copy was in storage under the bed. I don't know why.
Yeah, and the point is, if you do intervention more in some patients, it won't make any change because the problem is with housing and hygiene. So thank you.
Of course, thank you.